Okay, so welcome to the live practice session for the NPTEL course Environmental Biotechnology. This is an extra session which is intended to give a overall revision of the entire course. And I am Guru Priya, a Prime Minister Research Fellow in the Department of Civil Engineering, IIT Madras. So we have almost completed 11 weeks session so far, and in that few sessions had practice questions and few were just theoretical discussion. So in this session again, we'll just go by solving some of the questions from all the lectures that we have seen so far. So there will not be any theoretical slide or explanation as such. So whenever we see a question, we'll just see the explanation for the particular answer for that question. And mostly the questions would be of match the following, assertion and reasoning, and choose the best answer type. Just to have all concepts covered within this 2 hours of time. And in order to take this session forward, I would kindly request you to uh, participate in answering the questions that are displayed. So this is just a practice session, so need not worry if the answer is right or wrong. I anyways believe that the questions will really be easy for you to solve. And if you have any doubts from any particular concept from the lecture or any particular question that we'll be discussing now, you are always welcome to post it in the chat box or to unmute yourself and ask. So with that, I'll just display the first question for today, which is a match the following question. You just have to match the terms with their definitions. Sorry, Bhavya, then I am not sure. Maybe you can email the NPTEL support team and ask, because I could also read the rules only from the website as you are able to. So, okay, with that, the right answer for this question would be um, a guild is a metabolically related group of populations. It is not about taxonomy, whereas how organisms interact with each other based on their functions. So a guild is a metabolically related groups of population, whereas a population is a group of individuals of the same species in a same habitat. And a community is a set of all populations in an environment at a given time. So both in guild and community you have the term population. Population in itself means a group of individuals of the same species that are living in a particular habitat. And then you have a niche, which is a role or job a species performs in the nature. So you have to be pretty clear with the differences in all these terms. I would repeat that a guild basically means talks about only the function of different microorganisms, the relationship based on functions. Whereas in population, the keyword is the individuals of same species. Whereas in a community, you see that all populations that are present in the environment at a particular time. And niche, it's not about the individual, it's about the role or a job that the species performs in nature. I hope this is clear. So now we'll go to the next question. Again, I match the following question. So you have to match what are species richness, species abundance, mean, and other just two random things like 16 is rRNA gene and oligotrophic environment. Again, match with their definitions. Are you trying answering this or can I post the answers? Okay, so the answer for this question is again species richness is defined as the total number of different species present, whereas abundance is the proportion of each species in a community. So the difference between richness and abundance is the comparison. So in abundance, you just have a comparative analysis. So wherein you tell you have 100%, and in that how much of species A is present, species B is present, that is species abundance. So it talks about the proportion of each species in a community. Whereas if you talk about species richness, how many different species are present in there, that is about species richness, how diverse the particular environment is, how diverse the microorganisms in a particular environment are. So when you define this, that you call as species richness. And 16S rRNA, it's simple that it's a marker for microbial diversity studies. And when you talk about oligotropic environments, these environments are low nutrient and extreme environments. So species richness is how many different species are present. Abundance is just giving them a proportion or studying the relative percentages of each of the species. 16 as rRNA is a marker, and oligotropic environment refers to a low nutrient and extreme environment. So the next question would be an assertion and reason question. You can just give me if assertion and reason are right, or if any of the statement is false, and the reason is explaining the assertion. All these combinations are just how we have in a normal assertion and reason question. You can give any of the answer for this question. Okay, so this is a assertion and reason question. Uh, can you try answering this question? I'm sorry, I did not notice that I was on mute and I discussed the previous question. Uh, can you just start answering from this? And Bhavya, for your question, I think I answered it when I was on mute. I am really sorry about that. And I really have no idea on how this marking happens. I also have the information from the website only. So if you have any further queries, I would suggest you to write an email to the NPTEL support team so that they can assist you better. Okay, so the assertion here is in a microbial community, the most abundant species always has the greatest metabolic significance. And the reason is metabolic contribution is directly proportional to species abundance. And the answer is A and R both are true. Do you really think metabolic contribution is directly proportional to species abundance? We have seen some communities wherein though the microorganism number is low, the activity has been very significant that it could direct or orchestrate the entire community dynamics. So in that case, both the statement are false. So the reason for this is, let's say we have two species A and B. A is present in 100 numbers, for example, and B in 20 numbers. It is not necessary that the microorganism that is present in 100 numbers is going to be the key microorganism in the community that is directing the entire dynamics of that community or the system. if the species B is going to be metabolically very active, even though it is present in low numbers, if it is going to be metabolically very active, or if it is going to take up metabolism that is very essential uh, to drive the entire community. Like, for example, the starting point, let's say, this is just a lame example. Like, let's say there is complex organics somewhere. Hydrolytic organisms may be less in number, but they are very key. Only if hydrolysis happens, we can then go for any other conversion of that organic matter into any other product. So in that case, species abundance is not the deciding factor that tells us about metabolic contribution. It is not true always. It can be either ways. In some communities, the most abundant species can be uh, the greatest metabolic contributor, whereas in others, the one with very less abundance can still be the key metabolic contributor. So the next question is, which of the following best describes a microbial guilt? Exactly. So this is a right answer, and I had one of the questions here uh, where I talked about the same. So this was a match the following question, and we just saw. I'll just go through the question once again, which is um, we have to match between the terms and their definitions. So a guild is a metabolically related group of population, and when you talk about guilds, you keep in mind about the function of the microorganisms. And when you talk about population, the keyword here is the individuals of same species. So we call the individuals of same species that live in a same habitat to be a population, and a community is the set of all populations in an environment at a particular time. And when you talk about niche, it is not about microbes but about the role or job of the microorganisms that they perform in nature. So I'll reiterate that you have these definitions clear in mind. So whenever you see a question mentioning guild, population, community, or niche, you automatically figure out what that means and then go to the rest of the question. So this is the next question. In an acid mine drainage environment, which microbial characteristics can be mostly observed? Okay, so it is right. So acid mine drainage environment is going to be kind of a extreme environment, which is acidophilic environment, where there is going to be more acid. Not many organisms can survive here. So the species richness is going to be very low, and the abundance of few species is going to be very high. So when we talk about the species richness is total number of species that are present in that particular environment, right? So the total number of different species or the diversity of the species is going to be very less, because only the organisms which can survive such high acidic conditions can uh, be found there. So since the richness is going to be very low, abundance will automatically be very high, because when you count the total microbes as 100 percentage, the percentage of each individual species is going to be very high, because only few different species are present in that environment. So in acid mine drainage environment, low species richness and high abundance can be seen. High abundance of very few species can be seen. So next is a match the following question. Match the terms with their definition. So as I told, today's session will mostly have match the following question, assertion and reason question, and choose the right answer questions in order to cover most of the uh, lectures from all the 11 weeks. Okay, so the answer is right. Um, a community structure tells us about the taxonomic identity and abundance. So when we talk about defining the taxonomic identity and the abundance of microorganisms, we talk about the community structure. And in terms of phenotypic potential, we mean it is the potential metabolic capabilities. So, genotype and phenotype are two different things. Genotype is the genetic potential. Genes may be present but may not be expressed. When they are expressed, you call it as phenotypic uh, potential, and only when it is expressed, you can use it functionally. So, for metabolism. So, phenotypic potential is potential metabolic capability, and community function in the sense it is the realized metabolic activity in situ. So, when the microorganism is present live in an habitat, what metabolism it undergoes or how it contributes to the entire community. So studying that is community function. So the difference between the structure and function is when you study about the microorganism itself, like its taxonomic identity, its abundance, just about what is it and how many of it are there. That is community structure. When you talk about what is its function, what does it do actively, that is known as community function. And interrelationships, very easy. Interrelationships is the interactions among the species, and also the interaction of the species with its environment. Again, I match the following question to bring in all these terms into picture. Okay, so the answer is here. Superbug approach is an engineered single strain for pollutant degradation. So when you have a microorganism that is capable of some kind of environmental remediation, but still its capacity is can be improved, or its efficiency can be improved, then you go about engineering that microorganism. So you engineer the gene of the microorganism such that it increases the entire efficiency of environmental remediation that is brought about by that microorganism. So since you are engineering it and making it more efficient, you call it a superbug approach. And when you talk about microbial resource management, uh, it is exploiting the whole community's functional diversity. So you have different set of microorganisms which you term as the resource. The metabolic capability of the microorganism is in itself a resource for us to bring about some kind of remediation. So when we try to exploit the functional diversity of the whole community, then we go for this microbial resource management. So you have the details of which microorganism contributes to which kind of remediation, where you can use it, how you can enhance it. All this information you uh, get. And when you talk about metagenomics, it is sequencing all the genetic materials from an environment. Obviously, we have seen about metagenomics very extensively in many of the sessions. And when we talk about phenotypic microarray, it is high throughput substrate utilization profiling. So instead of growing the microorganism in different plates in larger quantities, you just have small wells where you add different substrates and a small amount of that microorganism, and check which substrate is more favorable for the growth of that microorganism. So that is a phenotypic microarray, which tells us high throughput in the sense it gives us uh, the ease of doing multiple experiments in a shorter time frame simultaneously, and the data that it gives is about the substrate utilization profiling. So these are all different approaches in environmental remediation. Again, we come back to some assertion and reason question. In a similar way, you can tell if whether both the statements are right, or if any of them or both of them are false, and if the reason perfectly explains the assertion. So you can answer it that. Exactly. So the assertion reads that environmental biotechnology processes are generally more economical and self controlling than physicochemical treatment methods. It's obviously more economical, and that's why it's obviously more economical and self controlling because the microorganism has its own mechanism to bring in this buffering action in the system. So the reason why it is more economical and self-controlling is because uh, when microbes are provided with essential nutrients, they can function autonomously with minimal human intervention. Because in a physicochemical process, you need some kind of regular monitoring and addition of some chemical, some kind of replacement, all of it is required. Compared to a physicochemical process, this intervention in a biological process is going to be very less. So it's true that it is economical and self-controlling, but yeah, it is just very slow and it has to be established further, and that is why we don't find so very established biological processes everywhere. We still have physicochemical processes in the system. Again, assertion and recent question based on the superbug that we saw a few minutes back in a match the following question. Absolutely, both are false. Superbug approach is not very much preferred because it involves genetic engineering and then setting up the strain, testing it, having the action continuous. So all of this is very laborious and time consuming, and it is yet to be established in a larger scale. The reason states that engineered strains are more robust. No, they are not much robust and sustainable in open environmental systems. The natural microbial communities actually are more robust and sustainable. It's exactly the opposite way. So, obviously, both the statements are false. Another question about biological nutrient removal. In which sequence does this BNR achieve complete nitrogen as well as phosphorus removal. Okay, so the right answer is aerobic, anoxic, and anaerobic. So, in a biological nitrogen removal process, which is the enhanced biological phosphorus removal or the A2O process, first you will have the aerobic system followed by the anoxic and anaerobic system. So, this is when all this nitrification and denitrification can happen properly uh, along with this phosphorus accumulation and then phosphorus release. So if you are going to change the order of this, the phosphorus accumulation and release will not happen properly, so you will not get complete phosphorus removal. So the sequence in which your biological nutrient removal system with nitrogen and phosphorus removal should be aerobic, anoxic, and anaerobic. Okay, I think this is a simple question for you. Exactly. So, metagenomics is about total DNA, proteomics is about protein expression, transcriptomics is about mRNA, and metabolomics is about the intermediates or metabolites that are produced in the metabolic cycle. Again, I match the following question with where you have to match the microorganisms with their role. Okay. So, dihalococcites, the term halo, you can always pair it up with chlorine. So, dihalococcites are going to perform reductive chlorination. Methanogenic archaea, again, it's very simple. It's about methane production. The confusion is between nitrifying and denitrifying bacteria. So, in case of nitrification, you're going to convert ammonia to nitrate. Whereas in terms of denitrification, the nitrate is going to be converted to nitrogen gas and be sent out of the system. So first nitrification happens followed by denitrification. So only in this part maybe you will be confused. Nitrification is the first step where you convert ammonia to nitrate followed by denitrification. So the next question is about the techniques in environmental biotechnology. Right. So high throughput essentially means handling multiple samples simultaneously. So and we always need high throughput techniques when we talk about environmental biotechnology because we have multiple microorganisms, different samples, high diversity. So a high throughput method which can handle many samples at the same time is preferred. When you talk about quantitative method, it's about measuring the abundance or activity. So you're not just telling if some activity is present or not, you're telling to what extent uh, the activity is performed. You define it numerically. Excuse me. And you define the number of species numerically, and that is a quantitative method. When you talk about structure function parallelism, it's about linking the taxonomy to function. Like when we saw about community structure and community function, in community structure it's about studying the taxonomy or the microorganisms present. In studying the community function, you will see what is the metabolic capability of that particular population. So structure function parallelism is you're comparing uh, the structure or the microorganisms that you have identified with the functions and draw conclusions about how the community is working. And in terms of PCR independent approach, it is to avoid the primer bias. I hope you remember what a primer bias is. When you have to do a polymerase chain reaction, the first thing you would need is a primer which can go and attach to the target sequence and then further extend and make copies of that. So if the primer is very specific and if it leaves out some genes, then you won't get copies of all genes you are looking for. So that missing out of few genes because you don't have proper primer for it is called the primer bias. And this happens in PCR based methods. In order to overcome this, you go for PCR independent techniques. Okay, we are again back to a assertion and reason question. in metagenomics, we only study about uh, the genetic composition of all the microbes that we see in a community. It is not going to give us any information about how the community is going to function. Only when you study about the expression of those genes, then it makes sense to define the function. So metagenomics cannot provide a complete picture of microbial community function. That is true because it only reveals the genetic potential. That is also true and it also explains. Are you able to figure out the answer for this question? Okay, we'll see the answer. Mm. A is true and B is false. As you told the reason, methanogenic archaea are very, very slow growing microorganisms. So they have a very low specific growth rate and they do not dominate the mixed culture. So you need to give them their space and time uh, to grow and produce methane. If not, they will get washed away. So that is what the assertion is explaining. You have to retain the slow growing biomass, in the sense microorganisms. You can rate it that way. You have to retain slow growing microorganisms for stable operation of methanogenic bioreactors. That is true. So in methanogenesis, methanogenic reactors, you have both bacteria and archaea. So comparatively, bacteria has a high growth rate than archaea, and the chances of archaea getting washed away if you are going to have a retention time based on the bacteria is very very high. So let's say if the bacteria can grow in 12 hours, the archaea can grow in 24 hours. If you have a retention time of 12 hours, then without growth itself, your archaea is going to get washed away from the system and you're not going to get any methane. So therefore, you have to retain the slow growing biomass by keeping the retention time a little bit higher to ensure a stable operation of methanogenic reactors. Why? Because this methanogenic archaea is a slow growing organism with a low specific growth rate, and does not uh, it is not very dominant in mixed culture. I hope the answer is clear now. Again, I think this is an easy question, like we saw about one of the match the following question about all the omics technologies. Exactly. So integration of omics data is very important to understand these both the structure and function of the community as well as its response and to predict its response to environmental perturbations or changes. So only when we know what microorganism is present and what is its metabolic capability, then we'll be able to predict if there is going to be a shift in temperature or shift in pH, how the system is going to uh, respond to it. So that is true, and for that you need both the information about the structure, function, and metabolism of that community. Each omics layer provides complementary information. So complementary information in the sense they support each other, and as a whole you get a strong connect connected story from which you can predict why a system uh, shows some such a behavior and how would it respond to any other change in the system. So both the statements are true and the reason explains the assertion. Okay. So you have to find out which of the following is not a limitation of conventional cultivation based method. I think this is one of the easiest questions. So when we talk about cultivation based methods, we have the problem of bias towards fast growing organisms and the inability to detect uncultivable species. And it gives only a limited representation of community diversity, whereas it can still do high throughput sample processing. So this is very comparative between all these four options. High throughput sample processing is not a very much high limitation of conventional cultivation based methods. So the next question is about what is the main advantage of integrating metagenomics, metatranscriptomics, and metaproteomics. I think this is kind of a repetitive question, so it's just that it provides a comprehensive view of the structure and function. So we'll go to the next question now, which is about uh, the different types of microorganisms. Okay. So lithotrophs are organisms that can use inorganic electron donors, and fungi uh, is mainly very much helpful in polymer degradation. Auxotrophs, they require external supply of growth factors, and endospore formation, it comes under adverse conditions. The microorganisms forms a endospore, which is a dormant state to survive adverse condition. Once the condition is back to favorable, then the endospore reverses and the microorganisms again becomes active. So right. Okay. Again, a basic question about microbial growth phases. This is kind of a very direct question. Exactly. So the answer is very straight here. In the lab phase, there is not much of growth. So the microorganism tries to get itself used to the conditions and nutrients of the system. Whereas in exponential phase, once the microorganism is set, it just directly starts growing in number and size. So you will see a maximum uh, multiplication rate and increase in cell number, while the substrate is going to logarithmically decrease. And in stationary phase, again, there is going to be depletion of the nutrients and accumulation of toxins, and hence you will see a flat line where the growth rate equals the death rate. And in the death phase, you will see irreversible cell damage. So, which means uh, there is high accumulation of toxins and severe starvation of nutrients. So, the microbe is no more able to grow and dies. So this cannot be reversed. That is irreversible cell damage and decline in viable cell count. So the number of live cells is going to be very, very low. So the next one is the assertion and reason question. So lithotropic microorganisms can grow in environments that lack organic carbon. Lithotropes use inorganic compounds as both energy and carbon source. So the term carbon source is the problem here, because when you have uh, oh sorry, I think I messed up with the answer here. It is both is true, as you have mentioned, both are right. So it can grow in organic carbonless environment. It can utilize inorganic compounds like carbon dioxide and other electron donors for energy and carbon. I'm really sorry, by mistake the answer was there. When I'm uploading the slides, I'll just correct that and upload it. So out of these four statements, which is the false statement? Right. So defined media is used for studying the metabolism of photolithotropes. Photolithotropes in the sense uh, they require light. A defined media, what is a defined media? It contains known chemical components, so we have precise control over the nutrient sources. There is no unknown component in that. And finally, the log phase in batch culture is characterized by a constant number of viable cells. It is not lag phase, it is the stationary phase. In stationary phase, the nutrient depletion and waste accumulation halt the cell division. This balances the growth rate and death rate. Therefore, you find a constant number of viable cells. So this question about the role of fungi. Exactly. So oxygenic photosynthesis is not possible with the fungi, whereas it helps in depolymerization, remediation of xenobiotics, and it plays a crucial role in composting. So regarding the different mechanisms in microbial metabolism, match the following. Right. So dissimilatory reduction means reduction of compounds with product excreted out from the cell. So you dissimilate from the system. Assimilatory means you are gathering it into the system. So the reduction with cellular biosynthesis is assimilatory. Syntropy is cooperative metabolism, where, for example, when three species exist, the first one tries to convert the substrate into a product A, second one then consumes A to produce B, and third consumes B to produce C. So this cooperation between these three organisms is syntropy, and terminal electron acceptor is the final recipient of electrons in respiration process. So here the assertion is whether anaerobic degradation of complex organic substrates requires the cooperation of multiple microbial species. Single pair cultures can completely degrade complex substrates under anaerobic conditions. So obviously both statements seemingly look contradictory, so one of it is going to be true and the other false. So exactly here the assertion is true, whereas the reason is false. In anaerobic degradation, we know that we have four steps, which is hydrolysis, acidogenesis, and methanogenesis, and each of it is performed by a significant group of microorganisms. So you need the cooperation of multiple microbial species. A single pure culture cannot perform all these steps. It can only one pure culture can perform only one of the steps, so you can expect a complete degradation of complex substances in anaerobic condition using a single pure culture. So again, one more question related to anaerobic processes. The accumulation of hydrogen during syntropic metabolism can inhibit the overall process. The reason is hydrogen consuming partners remove hydrogen, making the overall reaction thermodynamically favorable. Okay, so the answer is wrong here. So when you talk about this anaerobic metabolism, both in acidogenesis and acidogenesis, you have this hydrogen production. This hydrogen is further then consumed by methanogens to produce methane. Okay, so if this hydrogen is not consumed, then what happens is the hydrogen partial pressure increases, which is the hydrogen concentration increases in the system. So, to tell it in a very simple terms, you can imagine something like a feedback inhibition. There is some product produced. Only when that product is consumed, further the reaction will be possible in the forward direction. If the product accumulation is more, then the reaction that forms the product is going to be inhibited. So that is the whole concept behind this question. The accumulation of hydrogen inhibits the overall process because of high hydrogen partial pressure, more hydrogen production is not able to happen. So how this is rectified is hydrogen consuming bacteria consume the hydrogen and reduces the hydrogen partial pressure and concentration in the system. So this removes the thermodynamic limitation and then further allows more conversion of the substrate into hydrolyzed substrate into acid and hydrogen. So this is the concept behind this question. I hope that is clear. Okay, again, one more question related to dissimilatory reduction. Okay, I think there was a network glitch there. So I hope the screen is visible and I'm audible. The question is which of the following is not a characteristic of dissimilatory matter reduction? So in case of dissimilatory, you are going to send something out of the cell. In assimilatory, you are going to do biosynthesis. That is what we saw in the match the following question. So in case of dissimilatory reduction, biosynthesis is not going to happen. So usage of the components for biosynthesis or generating some biological biochemical compound is not going to happen. That is not a characteristic of dissimilatory reduction, whereas dissimilatory reduction involves sending the product out of the cell. It involves reduction of terminal electron acceptors and it is a key part of the catabolism. Okay, so the next question is about different techniques that we use in DNA studies. Oh, I'm sorry. Okay, so this question is about the different techniques that we use for DNA studies. I hope the previous question's answer was clear about the dissimilary reduction. Around 10 questions remaining now. So, can I just quickly go through all the question and answer randomly without waiting for you people? Is that fine with you? Okay. So, for this question, fluorescence in situ hybridization, it is used in the specific detection of taxonomic groups. So, just going to have a fluorescent tag in DNA and will be able to detect the taxonomic groups. Uh, in case of DAPI staining, it is diamidinophenylindole staining, and you do it to do the enumeration of total cell count. And enrichment culture, you use it for selective growth of functional microorganisms, functional groups in a sense, microorganisms. Quantitative PCR, you are going to quantify the gene abundance. And cardfish type of fluorescence in situ hybridization, where you do it for amplification of low abundance mRNA genes. So the next question is, can enrichment bias lead to overrepresentation of rapidly growing but minor microbial population? So they are telling that enrichment bias. So we are trying to enrich a culture, but only a favorable microorganism is growing, whereas you are leaving out few other microorganisms. So this can lead to overrepresentation of rapidly growing but minor microbial populations. The reason is laboratory media often provides higher nutrient concentrations than natural environments. This is true because in when you are going to cultivate a microorganism, you are going to supply the nutrients. It may not be sufficient for some microorganism which is very small in population but can rapidly grow. So you are going to miss out reading that particular microorganism. So hence both the assertion and reason are true. So what is a major limitation of fluorescent staining methods like DAPI or acridine orange? Uh, it can enumerate total cells, it can distinguish from live and dead cells, but it cannot provide phylogenetic information. It's just going to tell about the presence of a cell. It is not going to give you any phylogenetic information. You have to do genetic analysis to get this phylogenetic information. And obviously, we are using these methods on environmental samples. So the next is about the bioinformatics part. So 16S rRNA we saw already. It is a taxonomic marker gene. Binning is grouping the sequences based on their similarity, and contigs is contiguous sequences. So it is the assembly of short reads into longer continuous sequences. You have small fragments and you try to arrange these fragments in a particular order to get a longer continuous sequence. So when you do fragment based sequencing, you then go for contig formation. And rare microbiome is something that is of low abundance in a community. Again, one more about this bioinformatics, where metatranscriptomics, we all know it is about RNA. KEG mapper, it is a tool for mapping the genes to their metabolic pathway. Kyoto Encyclopedia of Genes and Genomes, that is what KEG stands for. KEG mapper, you use it for mapping the genes to their metabolic pathways. And then when you talk about primer bias, it is a systematic underrepresentation of the taxa due to PCR primer design. So this is what we talked about few slides back. You can't design primer that can fit for all genes, but if you miss out few key genes because you did not design the primer properly, then your analysis is not uh, very much valid. And when you talk about deep sequencing, it is the sequencing of community RNA to study the gene expression. So ideally, when you do gene sequencing, you see the genetic sequence, but when you do deep sequencing, you see which genes are expressed. You do the RNA analysis and see which genes are expressed and which aren't. When you talk about bioaugmentation, this is the last part of the environmental biotechnology course, and around week 10, 11 lectures we saw this. So bioaugmentation stands for introducing specialized microorganisms. So when you are going to supplement a system with microorganisms, you call it bioaugmentation. When you're going to supplement it with some nutrients or chemicals, you are going to call it biostimulation. So the key difference is when you're going to add microbes, it is augmentation. When you're going to add nutrients, it is stimulation. Terminal electron acceptor, that receives the electrons finally at the end of the metabolic pathway. Plate count anomaly we have seen since week 1. It is the discrepancy between microscopic and culture based cell counts. Not every microorganism is cultivable, so there is a huge bias in the study. And enrichment, so okay, we saw this question earlier, so I'm just skipping it. Which bacterial genus is closely associated with reduction of uranium? It is Geobacter. This is just a fact-based question. So we will see what is biosorption. Biosorption is a metabolism independent phenomenon. Just the metal is going to bind to the surface of the microorganism. So it is metabolism independent surface binding. Bioaccumulation is the microorganism is going to take the metal inside and going to accumulate it. So it is energy driven uptake into cell. So ATP is used here to take the metal into the cell. Bioreduction is the enzymatic conversion into insoluble form, and biomineralization is the precipitation via microbially produced ligands. So you produce some ligands, and when the microbe is going to attach to it, it's going sorry when the metal is going to attach to it, it's going to get precipitated. That is biomineralization. And when you talk about this metallothionein expression, it is going to enhance the metal binding or metal accumulation, and surface display engineering is reducing the cytoplasmic toxicity. So, because you can compartmentalize this metal uh, uptake into the cell. It can be either in the cytoplasm or it can be in the periplasm. So when you're going to have it on the surface only in the periplasm, then you're going to reduce the cytoplasmic toxicity that the metal is going to cause. Transporter engineering is uh, each metal has its own specific transporter. There are several transporters which are common for many metals. When you are going to engineer these transporters, you have a more efficient uptake and release of the metals to and fro the microbial cell. Consortium augmentation is the simultaneous removal of it is used for the simultaneous removal of multiple pollutants. So you have a consortium. If you're going to augment it with certain type of bacteria which can also work on one more pollutant that is present in the system, it's going to help you remove multiple pollutants. Sorry. So here biosorption is considered a metabolism independent process. Obviously, we saw it in one of the match the following questions. It relies on the presence of positively charged groups on the microbial cell surface. So the reason is false because microbial cell surface mostly has negatively charged groups. So here the key is the presence of negatively charged groups on microbial cell surface. If it had been this way, then both the assertion and reason are true, and reason would be the correct explanation of the assertion. When you talk about the presence of xenophobes in organic pollutants, uh, increases their recalcitrance to biodegradation. That is true. Xenophobes are structural groups uncommon in nature, making the pollutants less recognizable to microbial enzymes. So xenophobes are kind of uh, tricky phenomenon. It does not allow the enzyme to identify and do its work properly. So it is going to confer recalcitrance or resistance to breakdown by that microorganism. So this question is purely about different types of microorganism. You have acidothiobacillus peroxidans, which is very common iron and sulfur oxidizing bacteria. Acidothiobacillus. So acid and sulfur you have here, and iron peroxidants you have iron here. So iron and sulfur oxidation. Accumulibacter is polyphosphate accumulating bacteria. Anamox is Anamox bacteria. Anamox is anaerobic ammonia oxidation. So ammonia oxidation to nitrogen gas in anaerobic environment is done by Anamox bacteria, and Pseudomonas denitrificans is going to perform denitrification. So Anamox process is more energy efficient than conventional nitrification denitrification process for nitrogen removal because Anamox bacteria require organic carbon as electron donor. So the reason is false here. It does not require organic carbon as electron donor, and that is why it is more energy efficient than the conventional nitrification denitrification process. When we talk about this enhanced biological phosphorus removal, it requires alternative anaerobic and anaerobic condition because polyphosphate accumulating organisms store PHAs under anaerobic conditions and accumulate polyphosphate under aerobic conditions. That is true. That is what we saw in one of the previous questions for uh, nutrient removal, biological nutrient removal related question that was there before. So I guess the questions and answer was clear. If you have any further doubt, please post in the chat box, or we can close the session here. Thank you so much for your patient listening and your participation in today's session. I know that I was a bit faster by the end, or maybe when I upload the slides or upload the um, video, you can just go through it slowly. And we will also have the final session by next Tuesday. If you have any doubts, you can also come back by that time. From We'll have the next session, last session on coming Tuesday from 7 to 9. Are things clear? Do you have any further doubts? Sarita, do you have any further clarifications, or can we close the session? Okay, if there are no further doubts, we'll close the session now. Thank you for joining in. Hope to see you in the next session on Tuesday. Thank you.